Hello students, welcome to F.Y.B.Sc Microbiology class. Today we are going to discuss about the microscopy from the subject Microbiology paper 2 Basic Techniques in Microbiology. At the end of this lecture, the students are able to define microbiology, microscope, historical background. They will be aware with the structure and the function of compound microscope, how to use the microscope and how to take the care of the microscope. As we have discussed, bacteria they are too small to be seen by unaided or the naked eye, and therefore for their observation or to study them we have to make the use of magnifying system. The magnifying system which we are using is an instrument which can be called as the microscope. So, the microscope can be defined as optical instrument which consists of a lens or the combination of lenses so to make enlarged or magnified image of minute object. The microscope is a Greek word. Micron means small and the scopus means inch. So, microscope it helps for viewing the objects which are too small to be seen by naked or unaided eye, and the science that deals with the study of small objects using an instrument microscope is called as microscopy. Different types of the microscopes are there. First, we will discuss the classification of microscope depending on the number of lenses. So, first type of the microscope is the simple microscope, and second one is called as the compound microscope. Simple microscope they consist of only one lens and they are discovered by Antony van Leeuwenhoek. Now, this simple microscope they will differ from compound microscope which is having two sets of lenses. One is known as the objective and other is known as the eyepiece. In addition to the number of lenses, the second classification of microscope will be based on source of illumination. So, depending on the sources of illumination, they can be classified as light microscope, fluorescence microscope, electron microscope. Light microscopes are those in which the source of illumination is visible light, and they are also known as bright field microscope. While fluorescence microscopes are those where source of illumination is UV light. In case of the electron microscope, the source of illumination is electrons. Now, let's discuss the historical background of this microscope. In 1590, Hans Janssen and his son they developed the first microscope, while in 1609 the Galileo developed compound microscope which consists of combination of lenses. In 1620, Eugene redeveloped two-lens ocular system that was chromatically corrected. That is, it is free from any kind of the defects. Now, this is the microscope. Okay. So, first we will discuss about the different part of the microscope. This is light intensity control, light source. Next part is the condenser, stage, stage clips. These are the objective lenses. This is the nose piece, eyepiece, stage, coarse adjustment screw, fine adjustment screw or control, and this one is the light intensity control. We will discuss each part in detail. The bright field microscope, or which is also known as the light microscope or the compound microscope, is the most common and most dispensable instrument used in the biology laboratory. Basically, the parts of the microscope are divided into two types. The first part of the microscope are known as the mechanical part. Now, mechanical part are those parts which provide the structural framework to the microscope, and they will support the optical parts. While the optical parts, they are mainly involved in the magnification of the object. So first discuss the first mechanical part, base, pillar and the inclination joints. So the base, it is attached to the pillar, they will support the entire microscope. The base is generally horseshoe shaped or it may be sometimes U shaped. The pillar and the base, they are heavy so that to avoid the vibrations of the microscope. In addition to that, the inclination joints that will joins the arm of the microscope to the pillar is also present. And because of this inclination joint, the tilting of the microscope to any degree will be carried out. The next mechanical part is the arm and the body tube. Arm is also known as the handle of the microscope, and by using that we can carry microscope from one place to another place. The arm is bent solid piece of iron and that is attached to the pillar by the inclination joint, while the other end of the arm is attached to the body tube. The body tube, it is cylindrical in structure and at the point of attachment to the arm, graduated rack is there. And the main function of the body tube, it helps in the up and down movement of the microscope. Next one is the draw tube, that one resolving nose piece. Now resolving nose piece, it is fit in inside the upper end of the body tube and it it is at one end it is attached to the ocular lenses. Now what is the purpose of this draw tube? That is to adjust the tube length so that specified distance will be maintained between the objective lens and the ocular lens. Resolving nose piece, it is a circular rotating disc attached to the body tube and it carries objective lenses. Next mechanical part is the light or the source of illumination. In case of the compound microscope, the light used to illuminate the specimen from the base of the microscope. For this, you can make the use of low voltage halogen bulbs or LED lights are the most commonly used sources of illumination for compound microscope. So, what is the main function is to collect and reflect the light beams. There are two kinds of the devices which are attached to the base of the microscope. First one is the mirror or an artificial illumination. The main function of the mirror is to collect the light coming from a natural source or an artificial source and it will reflect the rays into the microscope. Now the mirror has both sides reflective, that is one side is plane and other is concave. Generally concave side is used when natural light is focused and plane side is used while focusing artificial light. In some of the compound microscope, built-in illumination provision is there and this device use 8 or 12-volt bulb and they will provide direct illumination to the stage. Light intensity control, these are the knobs which are used to adjust the amount of light that reaches the specimen or slide from the base of illumination. Next mechanical part, substage condenser and that iris diagram. So the light which was reflected by mirror or by inbuilt source will be diffuse and does not have sufficient density so to condense the light and focus it into the object. So this is the main function of the substage condenser, that is to condense the light and focus it on the object. So this consists of a condensing lens. The condensing lens it may be fixed or it may be loose in some microscopes. So the main function is to move up and down so that condenser will help to focus all the light on the object. Beneath the condenser, iris diaphragm is there which regulates the quantity of light entering into the condenser. So the iris diaphragm it may be closed or open with the help of a screw to regulate the entry of light. Next is fine and the coarse adjustment control. They can be used to adjust the focus of the microscope by moving the stage vertically. The coarse adjustment knob is moved to its highest position, and the fine adjustment knob is used to bring the image into sharp focus. Condenser condenses the light from the source of illumination and focus it into the stage. So it also have iris diaphragm which can be adjusted to match the effective numerical aperture of each objective lens. To open or close this iris diaphragm, the condenser ring can be rotated so that the amount of light which passes through it will match the requirement of objectives in use. Next mechanical part is the stage. It is the platform upon which the specimen or slide is placed, and it also has a hole in the center in line with the body tube, and the condenser below it allows the light to pass through it. In most of the modern microscope, the stage is fitted with a mechanical device called as mechanical stage, and it will help in vertical and horizontal movement of the slide. Next part is the stage clips or slide holder. The clips which are present on the stage, they can hold the slide in place on the mechanical stage. Next is the stage adjustment. By using this, the mechanical stage can be adjusted in the position horizontally in the X and Y plane. So to position the slide and proportion of the specimen is under the objective can be bring. These are the references. Thank you.